Welcome back to part 2 of this lecture. So when we left off last time, we were talking about this idea of an RNA world, saying that it may or may not represent what was driving life in its early stages. And now I want to talk in this lecture about prebiotic chemistry experiments that have been done to try to address life's origins. And I hope I can kind of frame them in the context of all the things that we talked about in the first half. And so I'd say this is what I call very crudely traditional prebiotic chemistry. I'm lumping together works from many, many people done in slightly different contexts over a period of about 70 years. So it's very crude and perhaps not totally fair generalization. But I'd say what most chemists working on the origin of life problem, or synthetic chemists anyway, have been trying to do is they've been trying to make biomolecules, the metabolites I showed you before, using simple chemistry, starting from simple C1 compounds like hydrogen cyanide or formaldehyde that are very reactive, that may have been existed potentially on the early Earth or not to large extents. What can I say about this? Well, sometimes they're done in the context of an RNA world. Sometimes these experiments were done decades before the RNA world even existed. So they use these high energy reagents, sometimes with light, sometimes without. And they eventually, if you change the conditions enough, sometimes these reactions occur all in one pot and you can get nucleobases just by mixing hydrogen cyanide together. In some cases, you have to carefully kind of craft the conditions and change them just at the right time or purify them just in the right way in between to arrive at complex biomolecules. Now, I think these works capture the imagination and skill and creativity of some really talented synthetic chemists. But what I'll question is whether they actually help us solve the origin of life problem. Now, why do I say this? Well, besides the fact that some of them require this kind of carefully crafted changing of conditions just in the right way, which kind of limits, I think, the plausibility of it happening, these ways of doing synthesis don't look anything like the biochemical pathways in a number of ways. So first of all, recall we learned that sugars, phosphates are made by gluconeogenesis in a very particular way. Here they're made by hydrogen cyanide and formaldehyde chemistry that uses different reagents, different types of reactions, different synthetic logic in terms of how the molecule is constructed. There's really no relationship at all to the biosynthetic pathways for the most part. In fact, some of these pathways were proposed because they couldn't get the biological pathway chemistry to work in the lab. It's also, I would say, largely incompatible with known biochemistry. Now, why do I say that? I think that might be a bit of a controversial statement, but hydrogen cyanide, for instance, is a super poison for metalloenzymes. Now, there's actually some metalloenzymes known to have a cyanide ligand on them, but if you add any exogenous cyanide, it poisons the metalloenzyme, and metalloenzymes are found to catalyze all those reactions in the core of metabolism, so not having metalloenzymes is a problem. Then there's the question of, well, if all of this chemistry was operating, then why did it completely change? Because it must have changed between here and the origin, last universal common ancestor, which is a very small time window, whereas in 4 billion years, nothing changed. So how could it all change completely at first and then no change for 4 billion years? It's another question. What are the mechanisms of that change? What are the selection pressures? None of that is addressed. And so, for all of those reasons, I have some doubts about whether this chemistry actually, just as beautiful as it is actually relevant to solving the origin of life problem. Okay, so now let's look at some of the models for the evolution of metabolic pathways. So how could metabolic pathways change over time? And this is kind of in a biological context for the most part. And one of the first hypotheses about this is the hypothesis of retrograde evolution, which came from Horowitz. And the idea here was there was a metabolite in the environment for which there is an enzyme that catalyzes a reaction to produce a product. So when I say an enzyme, that means there's already an evolutionary mechanism, there's already protein synthesis, this is already a biological system. And then, so that reaction exists, and then there's another metabolite that appears in the environment, or that it's already there, And then another enzyme can come in and catalyze the interconversion of that metabolite into the next one and then onto the product because that reaction already exists, and so on and so forth, eventually ending up with the biological pathway that we know today. So in a sense, you start off with the end product and the pathway over deep time evolves backwards to the starting materials. Okay? There's another hypothesis called forward evolution by Granick. And that is you start off with a starting material for which a reaction already exists, giving you the first product, and then that product is around now so that a new enzyme can come in and emerge and evolve, to catalyze that next reaction that gives you product 2, and so on and so forth. And in this case, your pathway evolves forward from starting materials to products in evolutionary time. Okay, so when I look at this one, what can we say? Well, you have to have the starting materials of basically all of these intermediates in this pathway have to already exist in the environment for this to work, right? And in this one, this is fine, but this kind of makes sense, but you have to have a lot of your initial starting material. That's fine. It could be something like CO2, but if it's something as simple as CO2, then how do you ever get the evolutionary mechanism to even do evolution in the first place or produce enzymes if all you have is this? So these could both potentially exist in biology, but they're not going to explain the origin of a metabolic pathway at the origin of life. You already have to have a developed metabolism and evolutionary mechanism and protein synthesis, etc. Okay, so here's another hypothesis, the promiscuous enzyme hypothesis. The idea is that, let's say you already have a metabolic pathway, and then a promiscuous enzyme appears that's able to catalyze many reactions, and it makes 3 different products appear from one central starting material. So it's what we call a divergent synthesis in organic chemistry. There's another hypothesis from Arturo Lescano and Stanley Miller, which is the patchwork evolution hypothesis. So let's say originally there was an ancestral pathway that existed. This could be organic chemistry, it could be biological chemistry. And then a new pathway emerges by incorporating reactions from metabolism into, from this pathway into the chain to create a new pathway that didn't exist before. Now for this to happen, you already have to have this starting material, an enzyme that it converts this into that starting material, and then something that converts the product into another product. So there's a question of where all that stuff came from. It already had to pre-exist, and why would a pathway exist if it wasn't interconnected? So there's nothing wrong with this model per se. Again, it could occur in biology, and probably does occur in biology, but I don't think it does a great job of explaining the origin of metabolic pathways in the first place. Another version that's more recent comes from Dan Taufik's, the late Dan Taufik's, late and great, I would say, Dan Taufik's lab. It's called the metabolite enzyme coevolution model. And in that version, there's non-enzymatic pathways that exist in addition to the enzymatic pathways. And over time, enzymes can evolve to catalyze the non-enzymatic pathway and make it more efficient and channel more flux through that pathway as opposed to another. And then, but that too requires already pre-existing metabolic pathways. So when we're talking about the evolution of metabolism before there's any pathways, it's hard to imagine anything other than having a full non-enzymatic pathway and then enzymes that come and start to catalyze it already, because otherwise you either run into a situation where you can't explain what why all the starting materials are already present in the environment, or you don't have any selection pressure for why an enzyme would evolve to do something. For instance, a pathway that has 10 steps. Not all of those intermediates are functional, right? So they don't necessarily add, some of them are functional, some of them aren't. So how do you evolve an enzyme to produce a non-functional product? It would either have to be one promiscuous enzyme evolving all of those reactions in one shot and then diverging, or simpler, you just have an entire metabolic pathway that's completely non-enzymatic, and that enzymes come in, that eventually produces enzymes. Those enzymes come in and reinforce the reactions that were already running. Okay, so this is the only model that I think really explains the origins of the first metabolic pathways. That does a good job at that. That doesn't run into one of those two problems. Okay, so the sixth and final take-home message is a good origins of life hypothesis should work in the lab, but it should also explain why life works the way that it does and not some other way. Synthesizing the building blocks of life is not enough, or whether they come from a miliary experiment or from a meteorite or a synthesis experiment. Synthesis within living systems is, like as I showed you in the first half, is seamlessly integrated into other aspects of dynamic self-organization, and it should therefore be part of a continuous reaction network that is self-organized. Most of the popular chemistry-based hypotheses don't satisfy those points above. Most models for evolution of metabolic pathways require that all intermediates of a pathway already exist in the environment. How does that happen? Or they don't provide a selection pressure when producing non-functional intermediates in the pathway. And a proposed solution to this is that the entire pathway started off fully non-enzymatic. Now, that is a big claim because we don't have that much evidence that they can start off fully non-enzymatic. And so that takes me to the kind of experimental part of this lecture. And you know, some of the, I'll try to condense what I'm going to show you into a manageable and understandable package. And so a lot of it comes from, you know, it's pulled from different people on this slide, or some of these people just contribute to this area and inspire me, although I don't show their work here. Some of them actively oppose what I'm about to say. I mean, Leslie Orgel, he passed away now, but he's written reviews that I think are critical of some of the ideas that I'll show you, but I'm happy to address any of those criticisms. And so I'd like to talk about that and give credit to those people. Okay, so if synthesis isn't the only way to solve the origin of life problem, how should we think about the problem in a structured way? And you know, if we draw on from other areas of science, we can ask the question, how does any self-organized system form in science? And so, you know, we can take one example, which is a hurricane, right? A hurricane is made of air and water, but air and water alone don't give us a hurricane. We only get the hurricane when we apply an energetic stress, and it has to be a very specific one applied within specific constraints. So for a hurricane, you have a temperature gradient that creates the self-organization, but not any temperature gradient will do. There's a certain magnitude that needs to be achieved. So if it's too small, nothing happens. If it's too large, you get chaotic behavior. It also needs to be applied on the right distance scale. And this is just a function of the underlying physics and rates of all the underlying processes. You get the hurricane behavior on the 1,000 kilometer scale, roughly. You don't get it when you apply that same gradient on the 1 centimeter scale or the 1 meter scale, right? It has to be on a 1,000 kilometer scale. So if we now think about the problem in those loose framework about how you get self-organized chemistry, you would need building blocks, right? So probably the geochemicals at the bottom of that pyramid slide I showed you in the first half. You would have to apply some sort of energetic stress. Now, based on how that pyramid works, I'd argue it's probably a redox gradient. It could be a chemical redox gradient. It could be an electrochemical redox gradient. It could be maybe a photoredox gradient, but I think that I would be cautioned about the third option just because it seemed to take roughly 2 billion years to evolve photosynthesis. So if all of self-organized chemistry that led to life was based on photoredox chemistry, then to me that would be a bit strange that then it took 2 billion years to evolve this mechanism again. But you know, could be wrong. There's other people that argue against that. And constraints, well, in chemistry, catalysts are important. They dictate whether one path happens versus another. They control the kinetic barriers to reactions, so they're probably important. There's lots of natural minerals that might be catalysts, so that could be a consideration. Scale, well, I mentioned the size of cells, right? 0.1 to 10 microns for the most part for the prokaryotes, so that's probably relevant. Conditions, things like temperature, pH. I hinted at electric fields potentially being important. This is not usually considered in this area, but I think it's time that we start considering it. And there's probably other constraints I haven't even been creative enough to think about yet, but we can imagine them existing. So in principle, if we want to create a certain type of self-organized chemistry, we can decide on a pattern that we want to see, and then we can do an enormous combinatorial search through building blocks, energetic stress, and constraints to search for conditions under which that particular pattern will emerge. So we then have to figure out what is the pattern we want to see, and we have to decide an experimental approach to searching through all those enormous criteria. And so that's what I'll, I'll mention in our strategy. But I just wanted to mention some ideas about self-organization before we get there. And it's kind of a general principle of self-organized systems that they emerge from the superimposition or simultaneous action of multiple simple mechanisms operating together. Okay, so a nice example of this is Conway's Game of Life, which was unfortunately, a game developed by the mathematician John Conway, who passed away, unfortunately, during COVID. And the idea was that he created a program with a series of time-dependent rules, almost like moves in chess. You know, the knight goes up one block and then 2 to the right. He could do time-dependent versions of that. So you take some black and white pixels, and if you have a pixel that's white, you go 1 pixel up, 3 to the left, and that turns black for 1/10 of a second. Okay, it's rules like that. And most combinations of rules lead to mundane behavior, but certain combinations of those rules lead to this very complex, almost lifelike behavior that you see here. And so it's kind of like finding a needle in a haystack. If you get the right combination of rules, really cool things happen. And so you can kind of apply that to self-organized chemistry. There's many chemical mechanisms that we know of in organic chemistry, but you can imagine there's some subset of those mechanisms that can operate together to give you complex chemical behavior. Of course, they also have to follow the laws of thermodynamics. It has to be energetically downhill overall. But the mechanisms of which what determines which mechanisms can happen is probably a function of the catalysts or the particular environment. So you can probably turn on or turn off some mechanisms depending on what environment you work in, and that's why some environments might give you this complex behavior and others not. Okay, so now what is the dynamic pattern that we should search for and try to recreate? And there, I would argue, based on writings of these 3 individuals that kind of motivated me to get into this field, that we should have something that resembles microbial metabolism, the stuff that I spent the first half telling you about, and all the bioenergetics associated with that, the compartmentalization, the whole thing, even the genetic aspect of it. And so this is a pattern we could imagine in a very simple form, obviously without enzymes at first, might emerge spontaneously. Okay, and this is a big ask, right? Because all of the reactions in this network are catalyzed by enzymes today and coenzymes. And we wouldn't have any of those to begin. So, but there might be something about the underlying pattern of reactions and the fact that they also come from a very small group of mechanisms, kind of like a self-organized system. If we look at the biochemical map I just showed you today, there's the same 7 mechanisms operating over and over again. That looks like a self-organized system. And so it may be that this emerged from self-organized chemistry. So then we could apply the logic I mentioned. We can search now for building blocks which are already kind of defined, energetic stresses and constraints, and try to find conditions under which this pattern would emerge. That is an incredibly complex task because unlike most experimental combinatorial searches that you would do in chemistry, this spans not just chemicals, but it also spans size regimes and the way that the energy source couples with the reactants and the type of energy source. So it's an extremely challenging combinatorial space to explore. So what I'll tell you about is what our lab has done to try to constrain that combinatorial space as much as we can so that we can search rationally within it relatively quickly. It's been about 8 years. I think we're actually coming to the end, I think, of that initial search. And I want to kind of tell you about what we did there. What we did is we actually went pathway by pathway and reaction by reaction within those pathways. And we tried to see how we could make it happen non-enzymatically, as many ways as we could. In that search, we found conditions that allowed some reactions to occur in many different conditions. So they're very easy reactions. That doesn't teach us much. It just means those reactions are easy. Other reactions we found to be very hard, and we only found one solution, or sometimes other people found the one solution to that problem. And those are very informative because they tell us, that constrains our search a lot. And so I'm going to now tell you what we did over those 8 years and how we've gone through that search. And I'm going to start with the pathway we learned about first, which is the acetyl-CoA pathway of CO2 fixation. So This is kind of the complex scheme I had drawn out before in the original slide. I've simplified it down now. So just to remind you that CO2 was reduced with 2 electrons to carbon monoxide. CO2 was reduced with 6 electrons to a methyl group via a formal or formate type group. And those combined to give us an acetyl group. And all of that happened on metals. Remember, it was nickel and iron and cobalt and maybe molybdenum or tungsten. And then there was a 2 electron reductive carboxylation with CO2 that happened on an iron-based cofactor to give us pyruvate. And so, this is a unique pathway for all these reasons I mentioned. It's thought to be very ancient, probably in the last universal common ancestor. And we wanted to see if this could be done non-enzymatically. And I'll summarize 3 studies in one slide. But basically, in 2018, we found that if you took just metallic iron powder out of the bottle and you put it in water and you added CO2, that you actually got all of the intermediates and end products of that pathway and almost nothing else. So we got methanol, we got formate, which are both C1, but more interestingly, we got acetate, which is C2, and we got pyruvate, which is C3, and we didn't see any other organics. Those happen to just be exactly the intermediates of those products if you hydrolyze them, of that pathway rather. So that's pretty specific range of products. We didn't get a Miller-Urey experiment with 300,000 compounds. We got just the intermediates of that pathway just by putting metal into water. So this worked over a wide range of temperatures between room temperature and 100 degrees Celsius. Above that, the pyruvate starts to decompose. Then after that, we published that paper. We got contacted by Bill Martin, who's a prominent evolutionary biologist in this field of early biological evolution and the origin of life. And he wanted to collaborate with us. He was super excited. He actually had been trying to do something exactly like this, but he's not a chemist, so it was a bit harder. And we decided to collaborate with him. What he wanted to do was to do another version of this chemistry, but rather than being driven by metallic iron, be driven by hydrogen gas, because that is what his modeling had shown the last universal common ancestor was breathing. Now, I don't know, there might be some, I don't know if that's universally accepted or not, probably not, but certainly hydrogen is one of the simplest and oldest sources of reducing agent in biology. And so we did an experiment together with this PhD student, Martina Preiner, who also came and worked in our lab. And she was co-supervised by Bill and by Haroon Toussouz, who's an inorganic chemist at the Max Planck Institute for Coal Research in Mülheim. And so Martina came to our lab and she joined 2 of my students, Srijith Varma, who was a PhD student with me, and Camilla Muhauska, who was a postdoc and eventually a CNRS researcher. And they worked together and they managed to come up with a version of this that worked using hydrogen gas. And so they were able to take simple minerals like the mineral awaruite, which is nickel-3 iron, it's an alloy, and it's found in hydrothermal vents where these organisms that actually breathe hydrogen gas and fix CO2 in this way live. And we found that if you just apply H2 and CO2, and now you heat to 100 degrees Celsius, a little bit higher pressures like you'd have a bit deeper down in the ocean, and then you could get the same products that we saw before with metallic iron, methanol, formate, acetate, and pyruvate. And then Haroon kind of took the project over, and he's more of an inorganic chemist. He was able to make nanoparticles of these sorts of mixed iron and other metal alloys. And he found that you could even do this at room temperature with these alloys using hydrogen as the fuel source, as long as the particle size was small enough. And so it does seem there is a non-enzymatic analog in its entirety, it seems, of this metabolic pathway. And it happens just with metal and water, or hydrogen and metal and water. So yeah, maybe this idea isn't so crazy after all. Okay, so now I get into the reverse Krebs cycle. Remember, this pathway is important because of the reason that it was making all the universal building blocks of metabolism, and it had this autocatalytic loop that can funnel matter and energy into just a couple of the intermediates that you need to build everything. And it was redundant with those 3 other metabolic pathways that more or less have the same function. Our thinking about this has changed over the years. What struck me was that of the 11 reactions in this cycle, 5 of them require ATP and 6 of them don't. So our initial kind of strategy was to focus on the ones that didn't depend on ATP because they'd be simpler. So that's what we did. We wanted to see if we could make these happen non-enzymatically as well. And this is actually chronologically the first project we ever did on this whole thing. And so we broke down the cycle into 2 chunks. One of those chunks was going from oxaloacetate to malate to fumarate to succinate. And the second chunk was going from oxalosuccinate to isocitrate to aconitate to citrate. So this is what we call a reduction reaction in pink, number 3. Number 4 is a dehydration reaction where you remove water. Number 5 is a reduction reaction where you add 2 electrons and 2 protons to this carbon-carbon bond, and you get the reduced form of that. And similar reactions are found here. You reduce the carbonyl to an alcohol, you remove water, and then you add it back on, but on a different carbon. Okay, so that's more or less what's happening. So we explored, we just went in the lab and just wanted to see how do we make this work. We didn't care about geological conditions. It's just about learning what works and what doesn't work. And so this was back in 2017 that we published this. We found that metallic iron, again, like the last pathway, does all the reduction reactions in the cycle. We found that if you went to really acidic conditions, especially if zinc 2 was around, you could catalyze these, well, I use catalyze loosely. It promotes these hydration-dehydration reactions here, and this one, chromium-3, seemed to do. So we were able to actually put these all together and have the reaction sequence go from here to the end and go from here to the end, all under the same conditions. So we were able to do 6 of those 11 reactions under common conditions. It just turns out the conditions that we found that do it are not very realistic, probably, for anything related to origin of life, because it was 1 molar hydrogen HCl, which is probably too acidic for any complex biopolymer to live in, although there are organisms that do live in stuff like this nowadays, but they're probably highly evolved. Anyway, don't take that too seriously. Later on, we started using hydrogen gas rather than metallic iron for the same reasons that Bill Martin wanted us to use hydrogen gas in the previous pathway. And we found that with heterogeneous nickel metal, that these reactions would occur as well. The hydrogen would be catalyzed by the nickel, and we could hydrogenate those reactions and allow that to happen. So think of this kind of like a hydrogenase enzyme. It's allowing the hydrogen to do its job. And that allowed that chemistry to happen at relatively mild pH. We could do most of it at room temperature and neutral pH. And then if we wanted to have the whole sequence happen, we had to go to pH 2 to get that to happen. So that was a milder condition, more in line with biology, but still missing these kind of linker reactions that depend on ATP and that fix CO2. Those are actually the hardest and most interesting reactions to look at. So we tried a lot of things. In fact, we had a Nature paper where we came up with an alternative solution to how to make all these things. I actually won't show you that paper today. We're doing aldol chemistry that life actually doesn't really do, but it ended up making a lot of the same intermediates of the reverse Krebs cycle, and I guess that's been our most famous paper, even though I'm not even sure how relevant it is. So much so that I'm not even showing it to you today. But what really blew me away was a very recent paper, only about 6 months ago, from a group in China, Zhang. And this comes in a long line of a sequence of papers that have come out over the past years from the groups of Richard Zare and Graham Cooks. Richard Zare is at Stanford, Cooks is at Purdue, both in the US. They're both analytical chemists, and they look at electrospray ionization mass spectrometry. And in that technique, you make these very small aqueous microdroplets. So tiny droplets of water that are about a micron apart, or even smaller than a micron. And one of the postdocs in Zare's lab got his own lab, and they had found, these two groups, over the last decade, that many reactions are dramatically accelerated by putting them in these droplets of this size. Sometimes 6, 7 orders of magnitude acceleration. And reactions that you would never even think to happen, like the reduction of dinitrogen into ammonia, happen in these droplets. So one of the postdocs from Zare's lab got his own lab, and he decided, I guess he'd read our papers, and he re-explored the reverse Krebs cycle reactions in these droplets. And he found that a lot of the reactions we couldn't get to work in our conditions, or that worked only under these harsh conditions, they work in microseconds in these droplets at room temperature with no catalyst. So for instance, he saw the carboxylation of ketoglutarate through to and its reduction through to isocitrate and its interconversion from isocitrate to citrate. So we've done this reaction using chromium and 1 molar HCl. Here it's happening in microseconds in the droplet. We never got this reaction to happen. And he also showed other reactions like reductive carboxylation that we never got to happen. They all seem to happen in this droplet under the same conditions. The retroaldol reaction that we hadn't got to work, that also works in these droplets. All the ones we couldn't get to work. And they happen in microseconds. So why do they happen? Well, some studies have been done to try to understand this. And so this is a little image of one of these droplets, and it's using Raman spectroscopy using a nanoparticulate pH sensor. So you can tell at the outside of this droplet, which is, here's a 10 micron scale, at the outside of this droplet where the droplet, it's very acidic at the edges of the droplet, blue here, that means acidic, low pH, and then it's alkaline in the interior, red. It's very basic. So this, is very unusual. Normally water is pH 7. Why is it pH 1 here and 13 there? Interesting. And this happens at the air-water interface where the droplet touches air. And at this size regime, specifically, that surface area is quite large compared to the volume of the interior. And by modeling this, and also through other experiments, it's been shown that there's a very strong electric field that forms at the very surface of this water droplet. This electric field has a distribution, but at the kind of tail of that distribution, it's almost as strong as the active site of an enzyme. Remember I said it was about 10 to the 9 volts per meter in the active site of an enzyme. And so this worked for many reactions. It was mostly done outside the context of metabolism, except for that one paper. And some other reactions I'll show you in a second, but I think this kind of brings our attention to electric field catalysis. So I'm not suggesting the origin of life happened in an aqueous microdroplet generated under electrospray mass ionization, but what I am saying is that electric fields, even without an enzyme scaffold, seem to be excellent catalysts, specifically for the reactions of biology. Okay, well, how about phosphorylation chemistry? We just saw a pH gradient exist in a droplet. It could probably exist in other environments as well. I showed you in the first half that one of the basic principles of metabolism and bioenergetics is that you have a proton gradient that collapses to give you phosphorylation chemistry. And so my lab, a postdoc in my lab stumbled across these papers, which I would have loved to have known about 10 years ago, but they've somehow stayed hidden in the literature, and they're almost never cited. In this paper, they did a very simple experiment where they took two aqueous layers. So one solution that's at acidic conditions, pH 0.3, so very acidic. Another that is at pH 10, so quite alkaline. And in the acidic layer, they put a molecule like succinic acid or fumaric acid or one of its derivatives, something that can form an anhydride. This is called an anhydride, the cyclic form of that eliminates water. And then on the alkaline side, they put phosphate. And they put a layer of chloroform that prevents these two layers from mixing. So they layer it and they stack it inside a tube so that the two things can't mix. And so there's a pH gradient from one aqueous layer to another that cannot be easily relaxed. And so in the acidic layer, there's a reversible formation of an anhydride. Once you form the anhydride, it's now soluble in the chloroform, so it can pass through the chloroform layer into the alkaline layer, where it immediately gets trapped by phosphate to form an acyl phosphate. So we've gone, now this happens as soon as you form the acyl phosphate, it gets deprotonated by the alkaline conditions, and so you've coupled phosphorylation to deprotonation, or to the collapse of a proton gradient. So with no enzymes, this thing is doing a job that's arguably harder than ATP synthase with no enzyme at all. It's phosphorylating something that's a better phosphorylating agent than ATP. It's doing it with no enzyme whatsoever, using just inorganic phosphate like the ATP synthase does. But remember, recall that the heat of hydrolysis of an acyl phosphate is minus 43 kilojoules per mole, and ATP is only minus 30, right? So this is actually harder to do than ATP synthase. Now, it requires a much larger pH gradient, 9.7 units, but this shows you that you can actually drive phosphorylation by collapsing a proton gradient, and this is done on a macroscopic scale. So later on, I'll invoke something that could happen like this, but on the microscopic scale. So very important paper. I don't think it's cited enough. It wasn't in the Origin of Life field even. It was just a purely chemical paper. OK, so now let's get into amino acid synthesis. How do you get out of this Krebs cycle into the amino acids? I mentioned to you there's two ways that happens. Reductive amination, where you add 2 electrons in ammonia, and transamination, where you shuttle that ammonia and 2 electrons around internally. And I showed you about pyridoxal phosphate being the coenzyme that mediates that process, along with the enzyme transaminase. And so the question is, can this chemistry happen without enzymes as well? And the answer is yes. So here's, oh, I should have put a slide on reductive amination. Transamination, we found, could be catalyzed by these simple metal ions. We showed it can work with nickel-2 or copper-2 or vanadium-5 or cobalt-2. Most of those are pretty abundant metals. Not as abundant as iron or aluminum, but they're not that rare. They're nothing like palladium or anything. And so we took this reaction between glycosylate and glutamic acid as a model reaction just to see how fast it could go. And with 20 mol% of the metal ion, here's the progress of the reaction over time with the uncatalyzed background reaction. And here it is with copper and with nickel. And with copper, it is 2,400 times faster than it is at 20 mol% loading compared to the uncatalyzed background reaction. And we can get multiple turnovers of the catalyst. So it's not just a promoter, it's actually a true catalyst that is not consumed during the reaction or bound up. So yeah, you can do transamination without enzymes, and we showed this works for the other keto acid combinations. We elucidated the mechanism. I took that out because I thought we wouldn't have enough time, but maybe actually we will. Okay, now let's go into sugar synthesis. So remember, this is happening through these pathways known as gluconeogenesis and the pentose phosphate pathway, which I showed you earlier. Can those happen non-enzymatically as well? Well, my lab has been working on gluconeogenesis for about three and a half years now, and we're just writing up our first paper on it, so I don't show you all the conditions that we use, but we have now done almost all but, well, I'd say all but one of these individual reactions in the lab in some way or another. Some ways are like this reaction we've done, but it's not really under conditions that are prebiotically plausible. We've published this in Chemical Science last year, but what it showed is that there's actually a mechanism to phosphorylate. So you might notice that this is not the phosphorylation of a carboxylate, like all the other phosphorylations I showed you in the core of metabolism. But what we showed you, what I showed in that paper, or what George showed rather, is that you can phosphorylate the carboxylate of pyruvate, and then it transfers the phosphoryl group intramolecularly to the enol form of this carbonyl, and that's how you end up at phosphoenol pyruvate. So the key kind of message in Joris's 2023 paper is that one mechanism of phosphorylation, the phosphorylation of a carboxylate, is actually enough to explain all the phosphorylations in core metabolism. Now, this is not the mechanism that biology uses today, but it's still the same overall reaction. So this may have been an evolutionary precursor to this, the mechanism that we see in biology today. All right, so Marcus Ralzer's group, he's a biochemist, and he was at the Crick, and now he's at Charité Hospital in Berlin. He had published in 2017 a paper showing that the non-enzymatic aldol reaction between glyceraldehyde 3-phosphate and 1,3-dihydroxyacetone phosphate occurs. This is when these 3 carbon units couple together to make a 6-carbon sugar. That occurs non-enzymatically, just in ice, spontaneously. And we've worked out the conditions for the other ones now more recently. So we don't have a condition that one condition that allows this whole pathway to happen, but I think we're at the point now where we can say each individual reaction is feasible non-enzymatically. I think that's the point where it's fair to say we can go. Now, Ralzer's also worked on the catabolic version of these pathways, the glycolysis pathway, and also the pentose phosphate pathway, which makes ribose 5-phosphate, which is actually the precursor to the building to how you make the nucleotides. Right? You make ribose 5-phosphate. It's phosphorylated to this PRPP, and that's actually what builds the nucleotides. He showed that much of this glycolysis pathway seems to occur just by mild heating with ferrous iron. And the same conditions can allow the pentose phosphate pathway reactions to occur. So if you stick, for instance, ribulose 5-phosphate in there, you can get ribose 5-phosphate out of it. It's relatively selective. It's not, I don't know how great the selectivity is, how they measure it, but it does produce this building block of the nucleotides. Now, there's also some early work done in the 60s about the non-enzymatic reactions in glycolysis as well, and by Mordecai Hallman, who was at the, I think in Israel, I think at the Weizmann Institute. So overall, it does seem like, even if I wouldn't say that we have conditions worked out to make all these things happen, that's for sure, but all of this chemistry individually seems non-enzymatically viable. Now, how about making the nucleotides from this? Okay, so our lab looked into this, and since I didn't teach you the pyrimidine biosynthesis pathway in the first half, I'll show it to you now. Hooray. I mentioned that it starts off from a very simple molecule called carbamoyl phosphate, which just comes from basically the ammonia embedded within amino acids, bicarbonate, and ATP. And that gives you carbamoyl phosphate, which reacts with the amino acid aspartate. That kicks out a phosphate and gives you carbamoyl aspartate, which then cyclizes to give you dihydroorotate, and it releases water. And dihydroorotate becomes oxidized. So this is a rare oxidation reaction. This is one of those 4 rare ones I mentioned. It's thermodynamically very easy, but not necessarily kinetically easy. And you get orotate. Now, orotate then in the pathway couples with this phosphoribosal pyrophosphate, or PRPP. And that's what gives you the initial non-canonical nucleotide, orotidine monophosphate. It gets decarboxylated, and that gives you uridine monophosphate. Okay, this is the actual canonical one that you use in RNA. Although first you have to phosphorylate it twice to the triphosphate before you can polymerize it. But anyway, this is the one we all want to make. Now, there is a salvage pathway that nature appears to have evolved that takes the uracil that comes out of the decomposition of UMP, and it recycles it by recoupling it to either phosphoribosal pyrophosphate or to ribose 1-phosphate. So those are called salvage pathways. But nature never makes this compound, it never makes uracil independently and couples it. It always follows this pathway. And I think this alone is evidence that there was not a pure RNA world that invented metabolism. Because if there was, when this decomposes, if there was an alternative chemistry that made UMP and it decomposes to uracil, there would have been selection pressure to evolve a pathway to make uracil. So we should see chemistry that makes uracil and couples this. But we don't. We see passing through what looks like self-organized chemistry through orotate, OMP, and then UMP. And yeah, this only makes sense if you have a self-organized chemistry that is not directed towards a certain goal, but certain functional intermediates turn out to be useful. Okay, so the question is, can this chemistry happen non-enzymatically? And so Jing in our lab looked into this. And she looked at all those individual reactions, and they all happen non-enzymatically relatively easily, with some caveats. Okay, so aspartate and carbamoyl phosphate, they just react together when you mix them at pH 8. There's no catalyst needed. And that forms carbamoyl aspartate in pretty good yields, in fact. Now this, under a variety of conditions, especially if there's copper-2 present, although it doesn't need to be present, but it can cyclize reversibly to give this dihydroorotate, which is the biological pathway. This is the biological pathway over here. But it also does a non-biological reaction. Well, I shouldn't say non-biological. This actually happens in biology, but it's not used to make anything. It also gives you the 5-membered product. So you can get 6-membered ring formation or 5. We see both. And that's not necessarily a problem, though, because when you oxidize the mixture of these compounds, dihydroorotate, when it's oxidized by manganese 4, it oxidizes to orotate. The tiny bit of uracil is a byproduct. But hydantoin 5-acetate, when you oxidize it, and then you expose it to alkaline conditions, it reorganizes and gives you orotate as well. So even if this channel initially existed, it may have been refined by evolution and the rates accelerated of this process simply because it's less things to optimize. There's only two steps here, and there's 3 here, so one less thing to evolve. Now, I'd be the first to admit that this is a bit of the same thing I accuse people of in the first HCN chemistry, of maybe being too controlling of reaction conditions. This can be done in one pot. We did it in one pot, but it does require human manipulation to change the pH at the right time, and that's cheating. So I think the thing you should take away from this is not that this chemistry is self-organized, but at least the individual steps are all feasible non-enzymatically. This is not chemistry that only an enzyme could do. It's chemistry that can actually occur without enzymes. Now, the next step, of course, is to take this orotate and couple it to ribose, right? To ribose 5-phosphate or PRPP. That's what the biological pathway does. We tried to do that, and it didn't work. And in fact, that was well known for a long time. Wasn't a surprise to us that didn't work. Leslie Orgel, who was a leading original life chemist in the 70s, already ran into this problem, and this was the inspiration for much of the work that was done by John Sutherland and other coworkers to make nucleotides in different ways. However, so is this the end of this idea of this being a non-enzymatic pathway? Well, the groups of Zaire, the same microdroplet techniques, they took ribose 1-phosphate, they put it in the nucleobase. Either they didn't take orotate, but they took uracil, which has a similar reactivity to orotate. They took also the purine bases, and they found that in those microdroplets, it forms the ribonucleotides. With no catalyst, no reagents, just the electric field coming from the droplet. So again, another, I think, line of evidence that electric fields may be important to catalysis of these key biological reactions. And our work that I showed you so far should only be taken as a proof that the reactions individually are non-enzymatically feasible. But we have to think about this sort of chemistry. So, and this happens in microseconds in these droplets. It's not a slow reaction. Okay. It also allowed phosphorylation to occur. So they took D-ribose and inorganic phosphate, and they were able to get the phosphorylated ribose. We don't know the extent to which this can work on phosphorylation more generally, but it seems very promising that electric fields could enable this stuff. Okay, so I'll summarize everything I've told you about the experimental studies that we've done. I forgot, I took out a slide on reductive amination, but we've also done prebiotic versions of reductive amination on these keto acids that mimic the way that ammonia enters metabolism. So here's all the papers that we've kind of done so far on this. One still to come. But I think we're to the point now where we've figured out that these reactions are non-enzymatically feasible individually. What seems to be, a lot of the reactions were easy. They worked a lot of different ways. But some of the reactions were hard, including the carboxylation, reductive carboxylation, phosphorylation, and ribosylation of pyrimidines. But those ones seem to work in these droplets, seemingly due to the electric field. So if I kind of extract all of the things, another reaction that was hard was the CO2 fixation to actually form CC bonds, and that only worked with these metallic iron and reduced metal complexes. So now if I can kind of, well, I'll come back to that at the end to answer the question of what was the likely environment where prebiotic chemistry actually kind of, well chemistry self-organized to form life. But for now, I just want to mention another entirely enormous problem as well, which is even if this metabolic network exists without enzymes, it's not enough. Because that network has to complexify or self-complexify to become something more complex like life. It can't just stick around producing building blocks. What we need to have happen is that the products of that network feed back as catalysts to reinforce the reactions that were already happening in the network, making them more robust, improving their selectivity. And that can prune the network, because if a branching point in a reaction could give you two products and you only catalyze one of those channels, now you make you prune the network in terms of the flux going through the network. But another thing that could happen is that you could you produce a molecule that has new chemical properties due to its structure that the previous molecules in the network didn't have. And it can have new reactivity, it can have new catalytic activity, and that will expand the network in different ways. So the network can change over time, even before there's, to a limited extent, but even before there's evolution, through a more traditional mechanism like genetics and protein synthesis. But can this actually happen? There's not much experimental evidence that can happen within the reactions of metabolism. So what we have done, I'll tell you 3 stories that I think are going to prove the point that this could happen. And they all involve coenzymes. That's why I told you about coenzymes. So I'll talk about the transamination reaction, because I already showed you a few slides ago that simple metal ions can catalyze transamination without the coenzyme and without the enzyme. And of course, today in biology, we know that that process is catalyzed by the coenzyme plus the enzyme. So you can imagine, if this is how things happen, that if the network starts off doing transamination, but then starts to produce a molecule like PLP that's a coenzyme that catalyzes transamination, that transamination gets better or faster or has some sort of advantage. And then that makes it more robust. And then at a later stage, the metal that was catalyzing it could be replaced by something that doesn't require metal, like that the network makes itself like an enzyme. Or actually, it's not been studied what happens to this transamination reaction under electric fields, so maybe you don't even need a metal ion at all. And in fact, you just make things faster and faster and less dependent on the original electric field that allowed it to happen in the first place. So either way, it allows you to get away from the original catalytic conditions that enabled this, maybe lessening your dependence on a rare metal, maybe lessening your dependence on an electric field, but you can escape the initial conditions for catalysis that your system needed to get started. Okay, so Quentin has looked at this, and we studied the transamination reaction between glutamic acid and pyruvate, catalyzed by either the cofactor alone, pyridoxal or pyridoxal phosphate, a metal alone, or both together. Okay, so here I show you, this is the rate of the transamination reaction as a function of the catalyst, and here you'll see the rate is a relative rate compared to the rate of the cofactor catalyzed reaction alone, so pyridoxal. That's set to 1, and everything else is relative to that 1. So copper alone, it's 11, okay, it's much better. Vanadium 5, 53, okay, much better. But the most interesting result actually is with iron and aluminum. So they're terrible, 0.5 and 2, terrible. Those are very abundant metals, but they're terrible catalysts for this reaction. Now let's look at what happens when you combine the coenzyme with the metal. Okay, so now you have copper 2 plus the cofactor, whoa, 98. So it is 98 times faster than the cofactor, the coenzyme alone, and it is 10 times faster roughly than the metal alone. But the most interesting result comes down here at the other end of this spectrum. These are ranked by the abundance in the Earth's crust, the two most being iron and aluminum. With iron plus the coenzyme, we see 90. This is 90 times faster than the coenzyme alone and 180 times faster than iron alone. This is a huge cooperative effect, and it's most pronounced with the most abundant metal. And the second best cooperative effect is with aluminum. So for some reason, the most abundant metals in the Earth's crust are giving the best cooperative effect, and really, and this is actually catalytic. It turns over. We showed there's 8 turnovers at least you can get with this. We never tried to push it further, but it is a true catalyst. So what I'm proposing then is that, okay, you start off with a non-enzymatic, non-coenzymatic transamination reaction, either catalyzed by a metal or an electric field. The network produces a small molecule that catalyzes this reaction, making it more robust or more selective, or less, and that allows it to occur in an environment that's now a bit different than the original environment, either having a dependence on metals that are more abundant or loosening its dependence on an initial electric field that allowed that reaction to happen in the first place. And that same kind of idea progresses on, where now you replace this initial metal with a catalyst that the network makes itself, and the network slowly becomes independent of the initial kind of environment that allowed it to start existing in the first place. So we're working now on finding out what is the minimal size of a peptide or protein that could be used to replace a metal here and outcompete this original process. Okay, now I'll move a second story, NADH. This is the most important redox cofactor for biochemistry. Here is its structure. It exists in the oxidized form NAD and the reduced form NADH. Okay, the electron carrier is actually this part, this nicotinamide moiety, And so it either is the positively charged oxidized form or it's neutral form where it's got this hydride donor. It can give a hydrogen plus 2 electrons. Now, this is, the main thing it does in metabolism is reduce carbonyls to hydroxy acids or reduce aminium ions to amino acids. So it's very important for that in metabolism. It's always mediated by an enzyme as well. And it's got another chicken and egg paradox. NADH is biosynthesized using the reactions that it catalyzes, carbonyl reduction and reductive amination. So those reactions must have happened before this could exist, and that's okay because we've published several papers showing that carbonyl reduction and reductive amination can occur non-enzymatically, as have other groups. So that's not a problem. But the key is that actually it had never been shown that NADH can catalyze or promote its non-enzymatic reactions without the enzyme. So you have to show that this can actually work without the enzyme, and then the enzyme comes later and makes things better. For that to work, you have to show this works without the enzyme. So we've done that now. It's true that, as other reports had written, that if you just mix the cofactor NADH with keto acids, it doesn't do anything, and there's no reaction. However, if you start adding metal ions in, we find that aluminum-3 and iron-3 both are the best at turning on the reactivity. Now, I think this is significant because it's also the same 2 metal ions that were the best for transamination with PLP. This is a totally different reaction, totally different cofactor, but it's the same 2 metals that fall out. Now, it turns out that since NADH is chiral, some of that chirality is transmitted to the product through the reaction mechanism, and we can get up to 60% EE, or an enantiomeric excess in this reaction. So that means that's roughly an 80 to 20 ratio of one enantiomer over the other. So it is possible to transmit chirality even at the level of a coenzyme before you even have enzymes around. So this may play into this question of the origin of homochirality. This is not a solution to this problem, but it shows you there are processes already at a very early level in non-enzymatic chemistry in which chirality can be relayed. And there's other studies showing this, but this is showing it in a metabolic context. So Robert, who did this study, Robert's now a junior group leader at the TUM and Technical University of Munich since January, but he's really good at DFT and mechanistic studies. And so he did some work where he lots of kinetics and computational modeling of the reaction. And we eventually came to the conclusion that what the metal does is it does 2 things. It binds the keto acid in a two-point fashion, which activates the carbonyl of this keto acid to make it better and more reactive. We call it a better electrophile. But it also binds the NADH through the ADP moiety. This is the diphosphate group of NADH. It binds it in a 2-point fashion. And that causes the hydride donor, the nicotinamide, to be pre-organized to deliver that hydride to the carbonyl. So it kind of pre-organizes everything just like an enzyme would do and makes the reaction a lot easier. It also activates it. And so he modeled not only this, there's actually 96 different stereoisomers of this complex, and he modeled all of them computationally, which is, it took a whole month of a supercomputer to do this. And he ranked the energies of their kinetic barriers, and the ones that are the lowest and easiest to get over, in fact, lead to the enantiomer that we observed, which is this R enantiomer. So the computations and experiments match, and we think we understand it pretty well. So that's a second example that I've shown you now out of those 3 stories I'm going to tell, where a coenzyme is able to do the same chemistry needed to make itself, and it depends always on the same 2 metals in both cases, iron-3 and aluminum-3. So those things could explain how our network becomes more robust, because as you form a molecule, it takes over a function that was already happening, making it better, and making it less dependent on the original conditions that allowed it to happen. But what we also need to show is that the network can grow and turn on new functions that didn't exist until the molecules in it were produced for the first time. And so I'm going to tell you a little story now about a very small, a very familiar metabolite, ADP. Right? This is the couple between ADP and ATP. Super important for phosphoryl transfer, right? Because it's the ADP to ATP reaction that's driving it's the phosphoryl transfer donor in all of bioenergetics. Okay, so it was discovered in the early 90s by a group of Japanese electrochemists by accident that if you take ADP and you have it in a reaction mixture, they had an old rusty iron electrode, but they figured out that it was actually the iron 3 that was on the electrode that was doing it. If you have iron 3 and you have acetyl phosphate, the phosphoryl group in acetyl phosphate gets transferred to ADP to give ATP. Now remember, the heat of hydrolysis of this is minus 41 kilojoules per mole. Here this is minus 30, so this is a downhill reaction, right? And you get ATP at room temperature in water. Now, Nick Lane's group, who's an evolutionary biologist at UCL, he found that, first of all, he reproduced the reaction. It works. He also, his student Silvana Pinna also found that when you change the nucleobase to anything else, it doesn't work anymore. Only ADP works, not CDP, not GDP, not UDP. They had tried a few other metals, not very many, but only iron 3 of the ones they tried worked. That's interesting too. And so we got interested in this because as mechanistic chemists, I wanted to know how this is working, and I also suspected that ADP was actually doing two separate things. It was acting as a substrate to receive the phosphoryl group, but it was also acting as a catalyst to mediate the transfer. And of course, we need the other nucleotide triphosphates because those are what we need to polymerize to make RNA. So we need that reaction to work for not just ADP, but for everything. So we imagine that if ADP is a catalyst for this reaction, we can separate those two roles of catalyst and substrate. We can use ADP as a catalyst, and now it can mediate phosphorylation of NDP using acetyl phosphate, and indeed that works. So we can now phosphorylate all the canonical and non-canonical nucleotides in water at room temperature, transferring phosphate groups from acetyl phosphate to the nucleotide diphosphates to give the corresponding triphosphates, which is what we need to get RNA. And you always get ATP as a byproduct of this process because you can't stop its own phosphorylation. It likes to phosphorylate another molecule of itself as much as it does any other NDP, and so you get that. The reaction also converts the monophosphates into a mix of diphosphate and triphosphate, so this can actually explain the whole set of reactions we see in metabolism, which is that you make the monophosphate, and then it gets phosphorylated to the diphosphate, and then the triphosphate. We're seeing the same reactivity non-enzymatically, just using metabolites. Okay, so we look at the metal dependence of this, and we did it in a more extensive way than Lane had done. And hey, look, iron 3, the best, aluminum 3, second best, nothing else works. Now we have 3 non-enzymatic metabolic reactions, 3 different cofactors, but they all depend on the same metals. I think this is pretty good evidence that those metals were around, or at least one of them was around at the origin of cofactors and metabolism. It's a pretty strong evidence. Okay, so I just told you 3 different short stories in which 3 different cofactors that are, I think, 3 of the most important cofactors in biochemistry mediate their non-enzymatic reaction either as catalysts or as reagents, but only in all 3 cases in the presence of iron 3 or aluminum 3. There actually has been a report since this paper is now kind of in minor revisions, but after we submitted it for publication, another group showed that you could do reductive amination rather than carbonyl reduction using NADH with no metal. So that also, that reaction can be done with no metal, but not the one we show here. Interesting, but I think that's probably not a coincidence, and we can probably put one of these metals in the environment where metabolism emerged. But more importantly, it shows that there are mechanisms for catalytic feedback and for discovery of new reaction and mechanisms through catalytic feedback in these simple networks. Okay, so the last thing before I end, and I think is the big question that we really all want to resolve, is in which environments can this chemistry co-occur? Right? We want not just the individual reactions, we want the real network, right? So you could argue the function of these whole 2 first phases of our research program was just to kind of get clues about what sorts of conditions would allow us to get the whole network to emerge spontaneously. And so here I'm going to present actually a brand new hypothesis that's not even published. So I hope you enjoy it, and I look forward to discussing with you later. It hopefully will be published by the time this video actually comes online. So if I was to summarize everything we learned from those 8 years of experiments and thinking from 2017, or well, 2016 really to now, and sum up the hard work of all the students and postdocs that worked on this over the years, what would I say? Well, we definitely need a redox gradient. We found that hydrogen gas can drive these reactions. We found that metallic iron can drive them. We showed that electrochemistry can, well, others have shown that electrochemistry can drive them. We're also doing some work on that now. We found the dependence on either iron-3 or aluminum-3, or maybe both. Those are essential, it seems, for cofactor activity, non-enzymatically. Iron-0 or nickel-0 are needed to reduce CO2 while forming CC bonds at the same time, both in the enzyme but also in the non-enzymatic work that we did. There doesn't seem to be an easy way around that to mimic the Wood-Lungdahl pathway. The iron can be produced transiently by hydrogenation, or you can imagine it could be produced by electrochemistry, but you have to form it at some point. It doesn't need to be sitting around in chunks, but it needs to be produced in some way. And a lot of the reactions that I didn't get really into, but they require either acid or base catalysis. So some of them need a bit of extra protons, some of them need a little bit extra hydroxide, but they need those to work. So, and then I showed you a bunch of reactions that seem to be like the bottlenecks for this whole process that were the hardest reactions that we found, but those seem to be promoted by electric fields, such as those found in the aqueous microdroplets. But I don't want to limit us to microdroplets. The principle of that catalysis is the electric field. So what I'll try to do, I think a good place for life to emerge would be, or for a self-organized reaction network like this to emerge, would be an environment that combined all of these things, or at least some of them, as many as possible in one place. So it has to have a redox gradient, metals, especially iron, in different oxidation states with a proton gradient. And we need a proton gradient anyway for phosphorylation and bioenergetics, and maybe strong electric fields for catalysis. So what environment could actually give us all these things at the same time? So now I have to talk about a hypothesis that is, I think, one of the leading hypotheses for the origin of life, which is called the alkaline hydrothermal vent hypothesis. And briefly, well, on the planet, right, we have metallic iron as the main kind of element in our core of a planet, which is very reducing. It's got a standard reduction potential of minus 450 millivolts, so it's quite reducing. And then we've got, when a planet forms, of course, the most dense layer is in the center, which is the metals, but the least dense layer is the atmosphere, the gases. And the water vapor in the atmosphere is constantly being hit by UV light and split into hydrogen and oxygen. The hydrogen escapes into space. We can actually see the hydrogen halo even today. So this is happening. So even without photosynthesis, there's already a minor mechanism to produce oxidants in the atmosphere. And that leads to a planetary redox gradient, reduced in the center and oxidized at the exterior. And the place where this is discharged is the hydrothermal vents and volcanoes. And so for that reason, this has been linked to the origin of life for some time. And I think Vectorshauser talked about it potentially in the late 80s. Mike Russell had predicted that there would be, famously, a milder type of hydrothermal vent that wasn't extremely hot, but that was more alkaline and a lower temperature. And later on, Deb Kelly and Jack Corliss did a deep sea exploration, actually found these vents that Russell had predicted. And so that was exciting the community. And then Bill Martin, who's an evolutionary biochemist and a biologist, he partnered with Russell, and very smart guy. He's my collaborator now, so I know firsthand. And him and Russell wrote a series of very influential papers that outlined how the core of biochemistry, including the acetyl-CoA pathway I showed you, including part of the reverse Krebs cycle, including amino acid biosynthesis, would emerge spontaneously under these conditions. And so the logic there is that as the reduced iron, especially iron-2, comes up and mixes with water, it does serpentinization, which is that it reacts with water to produce hydrogen gas and becomes iron-3. So you got your iron-3, you got your hydrogen gas. This is away from UV light, so you don't have kind of photodestruction of your organics. And the idea in those papers is that there is some sort of heterogeneous catalysis by minerals in the vents. And of course, our lab has published papers showing this is possible for these metabolic reactions, from CO2 fixation to amino acid biosynthesis to keto acid synthesis. There's also, because there's a temperature gradient, as the vent goes from the hardest point to the point where it meets the ocean, there's a temperature gradient between 2 and 70 degrees. So you can do this chemistry potentially at a huge range of temperatures. So If 70 degrees is too hot, not the end of the world. There was milder temperatures. And so they outlined that and of course there's organisms that actually live in these places that breathe hydrogen and do this chemistry. So it's not like you can't say it's unreasonable because it's actually happening today in those vents. It's just using biology. These are some photos that they sketched out from their original papers. And here are some images of the vents that they were interested in. Here's a schematic of how the gases and these reagents mix and react in these vents. And a lot of the reactions, here's the actual biochemistry that they mapped out. You can see this is very similar to the kind of pattern I showed them on my slide. It's not as extensive as that, but it's the same idea. And they speculated this would happen under heterogeneous catalysis. As I said, a lot of these individual steps, or almost all of them now, we've shown in the lab, or others have shown now, that can happen, but they're not happening in the same place under the same conditions. And so that's the real challenge I see from this, going from this hypothesis to something that actually works in the lab. That's the jump we have to make. So I'm going to attempt to propose what I think could help bridge this jump. Okay, so what are the strengths of this hypothesis? Okay, it's a known geological environment. I know both Russell and Martin reasonably well, so I can speak to the strengths and the weaknesses, I think. So it provides a constant, long-lived source of chemical energy, hydrogen gas, which is in the biological energy regime, is actually used by organisms today to do the reactions that are proposed. It's rich in reduced metals to act as catalysts, heterogeneous or homogeneous probably. It's shielded from UV, which I think is a positive thing. Other chemists might disagree. Organisms still live there doing exactly that chemistry by eating H2. And the conditions are in line with the experiments we have shown so far in the lab. Ballpark. I mean, there's quite a little, maybe I should remove this because there's a wide range of conditions in a vent. There's a wide range of conditions that we've shown, and not all of them are compatible with the vent per se, but they're mostly in line with this. Weaknesses. Well, one thing that's not really mentioned much in this is why phosphate in particular is so central to metabolism. So they do invoke reactions by which phosphate enters, but if self-organized chemistry puts phosphate at the heart of its metabolism, it should be because its chemistry is the easiest thing to do, not hard. And I would say under these conditions, there's no reason to believe that phosphate should be easy to incorporate into any reaction chemistry. So we have to explain that. And I showed you experimental evidence that's fairly recent that electric fields might help explain this, and that they're a central principle of biological catalysis. So maybe incorporating that into the equation would help. There's also no easy way to achieve reduction and oxidation reactions at the same time, even if those oxidation reactions are easy. I told you there's 4 of them that have to happen at least. And hydrogen is good at reduction, but it doesn't give a clear path to oxidation. And so we have to figure that out. I don't think this is really necessarily a criticism, but several people who are critical of the hydrothermal vent hypothesis have said it's too hot and too alkaline and too rich in metals ions to make RNA or have them sustain. But as I said, there's a wide range of temperatures that you can have in this vent, and also pH gradients, so we don't necessarily know those are true. But it is true that RNA is quite sensitive, and oxaloacetate and other metabolites fall apart pretty easily, so you have to synthesize them faster than they break down. So this is maybe a false weakness, but it's something we have to at least incorporate into our theory if we want to actually make it work in the lab. Okay, so here's what I propose as a kind of a modification to this hypothesis to kind of overcome some of these challenges. And it builds on 2 separate ideas. One of them is the discovery of geoelectric potentials at hydrothermal vents. So about 10 years ago, a group of Japanese geoscientists went down in a submarine, and they actually took a potentiometer with them. They measured potentials outside on the surface of these vents. And they found there was actually quite negative potentials on these vents. They found, it's hard for me to parse from what they've written exactly which one was a direct measurement and which one was a calculated measurement, but it seems there are potentials that are quite reducing on these vents. And they also showed that the vent material is conducting. So they actually took a light bulb and a battery, connected them by alligator clips, and the light bulb lights up, bridging the vent material. So So it's a conducting material. So electricity can flow through these minerals, and the reason that there is electricity is because you have hot reduced material and alkaline in the center, and you have cool oxidized material on the outside, and so there's a natural reduction potential gap from inside to outside. So the fact there's electrons popping out is not surprising. So that's one idea. Another idea comes from Mike Russell, who's kind of, like as I said, a leading figure in this hydrothermal vent hypothesis. But he's focalized lately on this idea of layered minerals such as fuggerite, otherwise known as green rust. And he says it's interesting, and I agree with him, because there are layers of iron oxides in mixed oxidation state between iron 2 and iron 3. And you can kind of control the ratio of iron 2 and iron 3 depending on the reduction potential. And then there's a layer of anions that sticks between them. These anions can be bicarbonate, they can be phosphate, they can be sulfate, they can be nitrate, they can be organic carboxylic acids. And depending on how big those things are, the size of those layers expands or contracts, and they've been known to span between 8 and 40 angstroms in the gap. So you can put organics up to 40 angstroms and as low as 8. And interestingly, that's the size regime where biosynthesis happens in core metabolism. Interesting. Okay, and all metabolites are anionic, so that could also explain how they end up in there. And that gives you your iron. So if you imagine now that you combine these two ideas and you have geoelectricity running through a mineral that's layered, you can kind of think of one of these layers as becoming like the anode of an electrochemical reactor, the other becoming like a cathode. And when you have that, you get a strong electric field. A potential like this, 0.5 volts over a distance of a few angstroms. Electric field is strong, should be 10 to the 9 volts per meter, like the strength inside an enzyme active site. You should have electric field catalysis like you do on the surface of that droplet. And it even gives you compartmentalization because the layers are acting kind of like walls, and you can only get into this mineral if you're negatively charged. So that also provides kind of a mechanism for selective entry of things inside and preserving them inside once they're there. So concentrating them, very much like a membrane does today. So these are both known ideas in the field, but I'm kind of combining them into one thing and adding electric field catalysis in there. And I think that could help allow a lot of these reactions that are enabled exclusively by electric field catalysis to occur in these layers. Now, how do you get out of that, and how does that explain biology? Well, first of all, if all of your catalysis is coming from, or most of your catalysis is coming from electric fields or from the reduced metals that are produced transiently by redox chemistry, that explains why most enzymes work by electric fields and transition metal catalysts in the core of metabolism. But it also can help explain proton gradients and their coupling to bioenergetics. Because on the surface of an electrode, the negatively charged electrode causes all the cation species to migrate out of solution towards it. So you don't get an equilibrium distribution of cations everywhere, like you would in a glass of water, but you get actually a non-equilibrium distribution where protons are on one side and hydroxide or whatever other anion you have, chloride, is on the other. And if this gap is small enough, like in the 6 to 40 angstrom gap like I mentioned, then small molecules only a couple of very small molecules can fit in here. Remember that a covalent bond is about 1 angstrom, right? So we're talking about molecules that are, 5 to 20 carbons big, right? That only those can actually fit in here and react. And they can take advantage of the electric field, but also the proton gradient that would exist in the space. And we already showed in principle how proton gradients that are stronger than 3 units can be used to directly phosphorylate carboxylates to acyl phosphates. That might explain why acyl phosphate chemistry is at the basis of metabolism. Yet ATP is the carrier. Now, why is ATP become the carrier? Because once you go to a weaker proton gradient, as this distance decreases, the electric field becomes weaker. You have weaker background catalysis from the electric field. You also have a weaker proton gradient. So you have less energy that you can use to harness. You need a weaker phosphorylating agent. ATP is weaker. Why is ATP chosen and not the other nucleobases? Because, as I showed with our experiment, only ATP is able to transfer or catalyze the transfer between acyl phosphate chemistry and ADP to form ATP. The other nucleobases don't do it. They can receive it, but they can't act to transfer it, and that's why I think ATP and ADP, that couple, becomes at the heart of phosphoryl transfer. Now, once you get into these larger distances, you still have a proton gradient, but you basically can't rely on the catalysis from the electric field anymore because it's not strong enough. So you can only survive in this size regime when you can make your own catalysts. And you don't need to rely on the electric field anymore. Then you go up to beyond a certain threshold, roughly the threshold of the size of a prokaryotic cell, and you can only, now there's no proton gradient anymore either because the gap is too large, and that can't really be harnessed in any productive way anymore. So to survive, you have to have a compartment that allows you to harness, you have to create your own proton gradient, basically. And so when you go to a place where there's a pH gradient less than 3 units, you have to make your own gradient to survive. And so you'd have to evolve some mechanism to make your own gradient like the electron transport chain. So I think this hypothesis is interesting because I think it is slightly more in line with what we know about chemistry actually working in the lab these days. But it can also help to explain why specific elements of metabolism work the way that they do and why electron transport is so key to all these processes. Okay, so with that, okay, I just want to give, from there, what are the big kind of things we could think about happening en route to life as we know it today? So basically, what I kind of showed you so far is steps 1 to 3, where if you have this electric field, it will broadly lower kinetic barriers, allowing many reactions to happen. Now, not all those reactions necessarily are in metabolism today, but this network autocatalysis could serve to prune some reaction paths over others, making sure the flux goes through them. And that's just due to the kinetic competition of having a self-reinforcing feedback loop. But then as the network produces its own small molecule catalyst, which I hinted through our experiments coenzymes appear to do, and there may be other things too that haven't been discovered yet, you get, you make certain paths more robust, you prune the network, and you make it less reliant on the original catalytic environment. So it can start to get more independent. Now, if some of these subnetworks can, so that's where we've kind of shown in the lab, at least in principle now. But you can imagine happening here is that this network also produces oligomeric uncoated polymers. So it starts to produce small polymers of peptides and small polymers of RNA. And those also, peptides can also act, and it's already known this can happen, as catalysts on those metabolic reactions. So they can start to catalyze metabolism. But if some of those peptides can start to catalyze RNA synthesis, which I think the code kind of hits at, could be true, and the RNA strands can start to catalyze peptide synthesis, then you would have a situation where if that catalysis is not completely sequence neutral, which it probably wouldn't be because chemistry isn't sequence neutral, you would start to develop, you would start to slowly transition from a completely uncoated production of those polymers to one that becomes coated. There'd be a link between the sequence of the polymers that are uncoated and feeding back on the synthesis of the other, and you could slowly get from a coated to slowly towards from an uncoated to a slowly coated regime. And so that would set us up for the relationship we see where proteins catalyze metabolism, RNA catalyzes protein synthesis catalyzes proteins catalyze RNA synthesis that we have at the heart of biology. Now, none of that requires RNA self-replication. That doesn't mean RNA self-replication doesn't happen. I'm not against an RNA world, but this is the thing that we have to explain the relationships that we see today, and RNA self-replication may be subsequent to this, but it doesn't require to set up this relationship. So with that, oh yeah, I wanted to say one more thing. So we also see interesting, I said some of these networks have to become independent. You have to make all your own catalysts. There's a concept that has been developed to explain this by Stuart Kaufman and developed by Mike Steele, Bill Martin, and Joanna Xavier. It's called reflexively autocatalytic and food-generated sets. So what the idea is that if you have a reaction network, it may contain inputs, which they call food, and metabolites, which could also be catalysts for each other's reactions. And you can get entities in which they are catalytically independent, where there's a catalyst for every other reaction in the network produced by the network. And when you get to that point, your reaction network becomes independent. It can exist anywhere because it's always producing its own catalysts. And so this is an idea that Kaufman came up with a long time ago, but Bill Martin and Joanna Xavier actually went through biochemistry and they considered the coenzymes and what they catalyze, and they showed that there's actually these sorts of rafts in metabolism itself. So I would suggest that at some stage of this process, you also develop, it may be before or after coding, I'm not really sure, but you start to generate these reflexively autocatalytic food-generated sets. And that's what allows you to get metabolic closure, to become independent of the catalysis of your environment. And exactly what stage that occurs is unknown, but I feel like it's probably a very important step. And then, of course, generating your own proton gradients would be the last, well, probably the last one, or near the end. Exactly what order these things happen is not clear, but I think they are kinetic selection parameters that have to happen at some point to get to life as we know it. Okay, so that's the end. I hope from these two lengthy lectures that the points you take away is that the chemistry that gave rise to life must have been self-organized. It was likely driven by redox agents and phosphorylation that was derived from the collapse of proton gradients. Much of core metabolism, I showed you experimentally, can happen without enzymes under metal or acid-base catalysis or electric field catalysis supporting this idea. Some metabolites, which we today call coenzymes, are ligands that accelerate catalysis by abundant metals. They reinforce existing reactions and they turn on new functions, and that can allow to prune or expand a network over time. And I've suggested this idea that electrochemistry on nanoscale, which I call nanogeoelectrochemistry, that could have occurred in vents, may have kicked off biochemical self-organization. So this could be a kind of modification of the hydrothermal vent hypothesis, leading to a cascade of kinetic selection processes that lead us to life as we know it. And so this is a new idea. We're starting to work on it in the lab, so we'll see how it evolves over the years. Probably it's wrong and I have to change it, but I thought I'd just present you how I think you can think about the problem in a structured way to organize your experiments. And I hope that was useful to you. I have to last thank the people that did the experiments, because I didn't do any of them myself. This is a photo from our lab reunion in 10-year lab reunion in 2022, where we got the alumni from across Europe to come back and have a party together. And we had a great time. They deserve all the credit for everything that's here. Collaborators, Bill Martin and Harun Tuzuz in Germany have been excellent collaborators. Funding is always the critical thing, and the ERC has been great. They gave me 2 back-to-back grants, and also the Volkswagen Foundation was essential as well. Thanks for inviting me, and I'm happy to take any questions.